Matt, like me, did not start his career as a biologist, and very unlike me, he actually is very capable of running companies. This is his second one. I used to get him to speak about the first one, but now he's going to talk about this one. Go ahead.
All right. It's an honor to be here. Thank you. And yeah, as Aubrey pointed out, in some respects, this is really the birthplace for me in the field of longevity. It's Aubrey who initially introduced me to this back when I was still the CEO of Immusoft at Sun 6 in Cambridge. And even Ocean itself got its start at one of Joe Becker's health extension salons and was run by Gary Hudson. So there's deep connections to everything here. And as Aubrey pointed out, my background is actually in the world of computer science. And I got into genetic medicine trying to build an app store for the human body. Or more technically speaking, an autologous long-lived plasma cell therapy that secreted therapeutics. And it's actually super exciting. That company is about to dose its first patient. But it was about 14 years from idea to this first patient, and probably about 12 since we'd gotten funded. And so my hope is that now there's thousands of genetic medicines in development, and back there were no approved cell and gene therapies. And so that the work we're doing today, and everyone's doing in this room, is hopefully going to be progressing into clinic faster. So we occupy a kind of a unique space in that Ocean itself is from day one dyed-in-the-wool longevity company, but we're mostly focusing our initial therapeutics on the health span side of things, and that's because we primarily operate in this world of FDA and big pharma, and so we've been putting together initial treatments to go after targets like this. We're based down in San Diego. If anyone is there, we have a beautiful lab. Cohabitate with a group called Entos, who developed a delivery technology that I'm going to talk about. Our initial focus is on a couple of the drivers of diseases of age, the loss of muscle and frailty, accumulation of unwanted fat, and as I think many of you are familiar, we got our start clearing senescent cells. Now, I'm going to tell you a little bit about how we're doing what we're doing before I get into actually the therapeutics themselves, because I think it's important, for one, and also this is something that I think can be applied to a lot of the work going on in here, and I'd like to collaborate. And so, I'll pick up a little bit where George Church left off at the beginning of this session, and about delivery of information into the body. And so people have basically wanted to mess with DNA ever since they knew what the double helix was, but your body has a vested interest in not letting that happen. And so today, at least in humans, the primary tools that are used to deliver genetic information are things like AAV, viral vectors, and LNPs. And both of these things have their strengths and weaknesses, pros and cons. Viruses, of course, are a purpose-built machine for delivering nucleic acids. They've had an awful lot of time to optimize this. They've been around for at least as long as life has. And, but your body knows what viruses are, and it doesn't like them. The AAV in particular has a relatively small payload capacity. They're expensive to make, and typically you can only administer them once. LNPs, on the other hand, are actually quite simple and inexpensive to manufacture. These were, of course, the basis for the famous COVID vaccines by Moderna and Pfizer. But they have some real issues in that they like to accumulate in the liver, and they become wildly toxic when they do. And so this has really limited the doses that you can administer with them. And I think as that technology initially emerged, people were a lot more optimistic about what you could do with it. And so basically what we've done is to try to take the best of both of these worlds. So using a delivery technology that's based on these lipids that you can make in a microfluidic system at scale and inexpensively, but using a viral fusion protein to enter the cell. And I'll talk about why that's important in a moment. But this technology was initially developed by Entos, and it had been obscure for quite a while, but in the last couple of years in particular, it's really gathered a lot of attention. Both because it took a COVID vaccine into phase 2 clinical trials, and also because they did a really large licensing deal with a big pharma. And so the thing that makes this PLV special is that it has a little fusion protein on it, referred to as a fast protein. And this thing originated from a reptilian orthoreovirus. It was initially studied by some virologists in Canada. And the virus actually doesn't even use it for cell entry. It uses it to fuse adjacent cells together to build a syncytia. And what's unique about it, or one of the things really, is that it is the smallest known fusogenic peptide sufficient for membrane fusion by a couple orders of magnitude. It's there for scale next to the HA protein from influenza. And you can see like that ectodomain is really tiny. It's only about a dozen amino acids long. And this allows it to effectively hide in the lipid particle itself and remain largely invisible to the immune system. It's one of the reasons we can redose this so well. And so the version we use, though, actually doesn't exist in nature. When the virologists initially characterized it, they went and screened the entire family of fusogenic orthoreoviruses and began to mix and match the different domains of their equivalent proteins. And then later did structure-function analysis and point mutations. And the version that we use now is vastly more potent than the natural ones. But what really makes this cool is that when a PLV gets close to a cell, it doesn't get endocytosed, doesn't need to be endocytosed like an LNP would. Those fusion proteins form a complex and directly mix the lipid of the particle with the lipid membrane of the cell. And so your cargo is delivered directly into the cytoplasm, bypassing the endocytotic pathway. And this greatly reduces the toxicity of them. And it also allows us to remove a lot of the lipids that were the most problematic in LNPs. So LNPs had to make a Faustian bargain of sorts with charge chemistry. And that since they need to be endocytosed to work, they must escape the endosome to deliver their payload. And so they have these conditionally ionic or cationic lipids that will effectively blow a hole in the endosome and get out. And this is really what mediates a lot of their toxicity when they accumulate in particular. And so this is actually an example of that. And I mean, one thing you may have noticed with LNPs is that they aren't being typically used to deliver DNA. They're almost all RNA-based. And that's something that I didn't notice immediately until we started actually using LNPs to deliver DNA. And they become really toxic. And they trigger, in fact, a kind of immune toxicity that was even worse than the damage they're doing to the livers there. You can look at that graph there on IL-6. I know it's hard to read, but this huge spike in IL-6 is caused from just half a mg per kg of the LNP. And that was the most we could give them and get them to survive long enough to test it. They're dead within 24 hours if you did a higher dose. But for reference, a dose 40 times higher of the PLV isn't even significant above background. And we think this is largely because it's exposing its payload to all this pattern recognition apparatus inside the endosome. And so your body has a vested interest in not letting you mess with its DNA, and it will examine these things and respond accordingly with the intracellular innate immune processes and then trigger extracellular ones. So it gets at really one of the big problems with LNPs. It's not that they don't work, they obviously work, but they're too toxic to use at high doses. And so if you want to be able to go after something as broad as the body, I would argue that none of those existing technologies will actually get us where we want to go. And so this is why I'm so excited about this particular technology. When you change all those lipids, you also radically alter the biodistribution. And if you're familiar with things like AAV and LNPs in particular, they love to go to the liver. And you can see here, the liver is down actually below the heart and the bone marrow. It's actually quite a bit different. And this is one of the early formulations that we built, and the data is in non-human primates, so IV administration. But now there are probably hundreds of these, I'd say, at this point, different formulations that actually have quite a bit different characteristics in terms of biodistribution. And like I said, once you were able to remove the ionic lipids from the equation, the parameter space we had to explore to choose new lipids and optimal formulations became much, much larger. So I said, we started in senolytics, and my goal at the time, like the perfect delivery technology I could envision, would have no tropism at all in a flat biodistribution curve. I wanted to be able to inject this into your veins and hit every cell in the body. And it's obviously a tall order, but this may be the closest thing that's ever existed to it. And I have this slide up in particular. It's actually a slide from our spin-out on oncology, Oncocenix. So it's a cancer example, but it really illustrates something that I was getting at, and it kind of ties back to my origins in computer science. I think how you approach the field, it governs how you think about it. So from my perspective, I looked at life as information, and I wanted to manipulate information in life. And so what we really wanted to do was to take all of the targeting out of the realm of chemistry and move it into the realm of information. Like I said, you get a vector that goes everywhere, but a payload that's specific. And the real, I think, profound advantage of this is that you can implement Boolean logic in DNA. You can put complex circuits in it. And this is effectively the control logic of life itself. You can do far more complex things. And so just in this example here, on the left, this mouse was injected IV with PLVs containing DNA encoding luciferase, firefly protein. And as you saw from the last biodistribution slide, it goes everywhere, the whole animal lights up. You sack it, chop it up, its organs are all lit up. But what's more interesting in this is the mouse on the right. And so that animal got the exact same dose, the exact same route of administration. So all those tissues have the same amount of DNA in them. But in that red dashed line, there's a tumor implanted. And now luciferase is under control of a synthetic version of the P53 promoter, something we're using to target cancer. And you can see that the expression is entirely restricted to the tumor. So this kind of turns the traditional delivery paradigm on its head. Like, it went everywhere, but it's only expressing there. And that, like I said, really allows you to do some interesting things. The broad biodistribution also allows for some very interesting routes of administration. As you can see, IV, it goes everywhere, but if you inject it into the muscle, it stays there. And that's also a really strong contrast to an LNP, where if you inject it into the muscle, in 24 hours, most of it will be in your liver. And the same goes for intrathecal, actually, or even like intracerebral. It will escape where the PLVs will stay. And so you can deliver all sorts of things. We actually have been playing around a little bit with oral administration of this too. But it gives you a great deal of flexibility, ranging from like what you might think of as an intermuscular application for making, say, those 3 genes that George Church was talking about, or going body-wide for a senolytic, or to make things widely across the body. Another really interesting thing about this is you can pair it with things like a nanoplasmid or a mini-circle, and you can get very durable expression from a non-integrating vector. So that's what we're seeing here. And first of all, I guess I'll point out just how deep it's penetrated the tissues. So we're now staining for luciferase, so you can see it in IHC. But we're basically hitting every cell in the tissue. And we're getting expression, in this case, it's for a couple months. And we've seen it for like around a year, I think. And so you're getting this durable expression without integration and really broad tissue penetration. You also, despite my general philosophy of wanting to target with information, can attach actual targeting moieties to this to bind to surface receptors. And it has an added benefit in that since the surface receptor doesn't need to be endocytosed, you can hit pretty much anything you can see on the cell. And this is facilitated by an interesting quirk of manufacturing in that these things are made in a single step on a microfluidic system. In that fusion protein, when it's inserted in the PLV, is upside down half the time. And that doesn't really cause any trouble. It just doesn't work when it's upside down. And so you can actually attach a targeting ligand to the inside of it, and then half of those end up on the outside. And so you actually don't need an additional step or another protein, which I think is super cool. Finally, like I said, with respect to the platform, since this thing is effectively invisible to the immune system, you can administer it multiple times. Here, in mice for the better part of a year, and in monkeys, even with repeat dosing, you never see any kind of neutralizing antibodies against it. In fact, it took the lab that was first studying this several years to deliberately raise an antibody against it. So even if you inject it with adjuvant, you don't get any kind of neutralizing response. Okay. So now on to some of the fun stuff we're doing with it. The first therapy that we're taking to clinic is to make you effectively build muscle without exercise to treat frailty and sarcopenia with age. And the reason this has really become the lead indication for us is because pharma likes it. So one took an option on it about a year ago. And so we have prioritized this. And fortunately, the genetic levers that govern skeletal muscle growth are actually pretty well understood. So that monstrous-looking cow you see there on the left, it's called the Belgian blue cow. It's first documented in the early 1800s. It has a defect in a gene called myostatin, a negative regulator of muscle growth. So classic phenotype of this is that it's about twice the muscle mass of its wild-type equivalent. There on the right, you see some work in mice where they have overexpressed follistatin on the far right, in a transgenic animal, so from birth. And the phenotype for that is 4 times the muscle mass, so it's absolutely enormous. But if you administer these later in life, they do not get that monstrous. But the point is that this has been studied for a long time, and the pharmaceutical companies have been interested in it for a long time also. And everything from making antibodies against myostatin to follistatin fusion proteins. They viewed this as a very promising target, but have never really been able to overcome the CMC issues to manufacture it and turn it into a drug. And so we think that we've managed to solve that. And so the reason you want to do this is as you get older, physical strength begins to count for more and more. I mean, you can think of in an extreme case, from the moment you end up in a wheelchair, you're on borrowed time. Like, if you don't get out quick, you won't get out at all. But muscle also provides a lot of protection against things like your joints and your bones, and helps you regain balance if you slip. And so the idea of making someone stronger, more active, and less pain is really, really attractive to us. And it's a little demoralizing for someone who works in genetic medicine, but it's devilishly hard to beat regular exercise in terms of health span improvements. And so being able to enable people to be active is very important. So what we did here was we injected these mice, also tail vein, single administration, the PLVs containing nanoplasmid encoding follistatin. And as you can see here, in about 6 months, they were 50% larger and twice as strong. And so this goes to that durable expression. It's not integrating, it's just making it continually. And we've put this in primates. There's actually been human gene therapies using follistatin with AAV. It's, I think, generally viewed as safe and efficacious. And I think that we have the ability now to manufacture it at a scale and cost that would be accessible to basically the entire aged population. You can also administer this locally, interestingly enough. So the last ones are IV, because we want to make the whole body strong. But if you inject it directly into a muscle, that muscle gets meaningfully larger than its contralateral one. And this has applications, of course, practically, but also cosmetically. And I say that because farmers might be willing to pay for it. And if you can make a lot of money doing it and you show that it's safe, that's great for the space also. Probably the newest program we've been working on is actually killing fat cells. And this is actually based on the same general technology we use to kill senescent cells and cancerous cells. It's basically driving a caspase in an adipocyte targeted construct. And the first version we've built for this is designed for local administration. But our goal with this a bit longer term is to modify this payload to target only visceral fat. So you could inject it IV, ablate your visceral fat, and not hurt anything else. I think this has a lot more consequences in metabolism and age and inflammation. A little bit of history. Some, I'm sure, in this audience know, but what got Ocean started was actually this senolytic data with the ink attack mice, where you could selectively delete P16 positive senescent cells with an otherwise innocuous drug. But what I didn't realize at the time was that wasn't the original model for that. It was based on one called the fat attack model that was designed to kill adipocytes. And they could cross this with these super fat mice and ablate every living adipocyte in it in 3 days, and which I do not think is good for human health. But it was actually really amazing, and I think kind of fun that it ties all these programs back together. Our first target for this is actually a disease called lymphedema. It's a rare orphan disease. It primarily affects women. It's characterized by abnormal fat deposition in the trunk and lower extremities. And it's technically a connective tissue disease, but current standard of care is lifelong ketosis and repeat liposuction. So it's really nasty. And I thought we can turn that into a simple shot as opposed to like all the trauma of liposuction and ablate the cells locally and show safety and efficacy there. We've done some proof of concept with this. Like I said, this is an earlier program for us, but we started actually by taking human tissue explants. So these were excised from tummy tucks, and they would basically make little cookie cutter punches of this and send them to our lab. And we could inject full thickness human skin with the PLVs and see how we're killing the fat and look for other damage. And so the first iteration of this, which is shown here, was killing around 20% of the adipocytes, but it wasn't causing any damage that we could see histologically. And so this kind of launched a campaign to optimize constructs and formulations, and that's what we're going for with it now. Okay. So I want to wrap up. So we have like really big or really broad rights to this delivery technology in the field of longevity. And like I said, we've focused on a few things. Initially, we actually are too small to go after all of them. And so we are super eager to collaborate on things that people are working on today. And I think a lot of the treatments that have been elucidated through their genetic component are often kind of tried to be developed into drugs with small molecules. And that some of that might be able to be short-circuited entirely with a genetic medicine. And so we always love to kick around ideas. Please come talk to us. Some of our senior team, they deserve a lot more credit for this than I do, and are also happy to chat about any brilliant ideas you may have. Thank you.
Brilliant, thanks, Matt. Honestly, mind-blowing technology, isn't it? Questions? Over there. Microphone, please. Thank you.
Thanks. Sorry, it might just be a part of your animations or your drawings, but your structures look more like liposomes than LMPs. So they're fluid-filled, rather. LMPs are filled with carriers and other lipids.
Yeah, so they're you have helper lipids to like initially encapsulate the nucleic acid payload, and then the outer lipids on it as well. So it's closer, I'd say, to an LNP than anything else, but it's a little bit different. And I won't get in too far over my head, and I'm not the brilliant lipid chemist behind all this, but yeah, it's I think relatively tightly bound up with its helper lipids.
Yep, there's another question. Microphone's coming.
I always enjoy your talks, Matt. Can you tell us, or just sort of speculate into the future about the kinds of complexity of logic that might be possible, like not just simple additions, but maybe more Boolean possibilities?
Yeah, well, we've gone down the rabbit hole with this. Like I said, even in the visceral fat construct, it turned out to be more challenging than we gave it credit for, in that there wasn't like an obvious promoter you could select that was on in just visceral fat and not in subcutaneous, and that would be safe enough to ablate across the body. And so in that version, you start having to look at like the relative levels of promoters and repressors. And I think it really, at some point, this blurs into synthetic biology more, in that as you start screening, transcription factor binding motifs and enhancers and repressors across the genome, you may well end up building what amounts to a completely de novo promoter based on things that are in human biology but that don't exist in nature. Probably the most extreme one I've played with was an optogenetic circuit, where I wanted to be able to make a drug on demand. So you could inject this into your muscle, it'd be responsive to like infrared light, and you could turn it on and off at will. And so it allows you to pulse therapies. And these are super hard because there's definitely no mammalian equivalents to them, and they also require things like amplifier circuits. So I think like, in general, there's going to be a marriage between synthetic biology and computer science with medicine, and that's going to be the future of the space.
Great. Question right in the middle. There you go.
Thank you for your talk. So regarding lipedema, how selective can the therapy be? to find the dysfunctional adipocytes? So how dysfunctional, how selective does it have to be, and how selective is it at the moment?
It's actually just killing all adipocytes. So the disease is more of a, their abnormal deposition than I think the genetic of the adipocyte itself. And to be fair, though, the etiology for it is largely unknown. Like, there isn't a good animal model for it, for example. And so this, you could think of, in some respects, as liposuction without surgery or trauma. It is going to just ablate the adipocytes and ideally nothing else, but it won't differentiate beyond that.
But you would assume that you could make a second-generation version for diabetes, for example, that was only hitting visceral adipocytes.
Yeah. So that's where we're going with this. And but it's always this trade-off of what can you build today, what will the regulators tolerate, and making money.
All right. No more questions.
Oh, sorry, just one more question. Yeah, does follistatin operate in a much safer way in comparison to something like corticosteroids and muscle gain?
Yeah. It's actually also not sex-specific, so it's the exact same in both. And that's one of the beauties of it. All the anabolic steroids have this mix of androgenic and anabolic effects. The follistatin is basically the molecular pathway for skeletal muscle exercise. And so when you exercise your muscles, you damage them, they call for help, the liver hears their pleas, and it creates follistatin. And so this is your actual biochemical pathway, and we effectively just jam it on 24/7.
All right, thank you.
Thank you.